I'm going to be speaking about water, and I guess the first question that you might ask is, do we really understand water? And I think most of you would say, if you're outside the field, of course we understand everything there is to know about water, because it's such a simple molecule. It's been studied for hundreds of years by many scientists. And in the next few slides, I'm going to demonstrate to you that that's completely untrue. So here's a question for you. Clouds are made of droplets of water. Droplets of water are heavier than air. How come they float? And if you can't answer that question, then you probably don't know all there is to know about water. Here's another example. You take two beakers that are essentially full, and you stick two electrodes in, one in one beaker and another in the other beaker. And here's what happens. Forms a bridge. And if you move one beaker mechanically away from the other beaker, the bridge persists. And it can persist for a distance up to 3 or 4 centimeters, and essentially indefinitely. So please, can you explain it? I don't see any hands going up. And if you can't explain it, you don't know all there is to know about water. Now, another one that's more familiar to you is drop some droplets of water on the surface of water. And of course, you know that the two coalesce instantly. However, it's not always true. You can see what happens. This is a demonstration from our laboratory. Please, can you explain it? An explanation. Okay, surface tension. Okay, well then I yield to you. So that one is understandable. I think that's not exactly the answer, but good for you. Okay, so here's another one, and you might explain this one the same way. This is a lizard that comes from Central America. It sits on a branch, and by the way, this is taken from National Geographic. Sits on a branch, and when it gets inspired, it jumps from the branch and basically jumps in the water, and it walks on the water. So I don't think you can walk on the water, or maybe you can, but because it walks on the water, it's called the Jesus Christ lizard. It needs an explanation. Well, so here's one that is my favorite. This is a container, a trough of water, and some red dye is put in the water for reasons that will become obvious in a moment, but it has nothing to do with the phenomenon. And behind it is a superconducting magnet. You turn on the magnet, and the Red Sea splits so that Moses could walk across it. This is for real. I saw a demonstration of it. So please explain it. I don't see too many hands up. Okay, this is a problem if you're a fisherman. It's hard to catch the fish. Okay, so anyway, I was in a different field 2 decades ago, and I came upon this guy who was my great inspiration. His name is Gilbert Ling, and he was chosen among all young Chinese scientists in 1948 to come and study in the U.S. He was one of 3, and at least one of them won a Nobel Prize, and my Chinese student tells me the second one won a Nobel Prize. And he contributed so much that, one would think that he might win a Nobel Prize. And his contention was that water in biology, especially water inside the cell, was not like this glass of water, that the water molecules in this liquid water, the water molecules are randomly disposed and bouncing around a fierce number of times per second, or even per femtosecond. And he said, no, it's like that, not like that. He said the water molecules are organized in a way, stacked one upon another. He had a lot of evidence for it. I met him at a conference, and I heard his evidence, and I heard evidence from people who had experimental data to support his contention. And I was mesmerized by this. I thought, this is so interesting. And so I gave one of his books to my students, and uniformly their feedback was, if this guy is right, and it looks like he's right based on the evidence, all of biology is wrong. Because all of biology is based on the contention that the water inside the cell is just like ordinary liquid water. And he said, no, it's not like it. So we had no money to study water. We had some money to study mussels. And so I was thinking, what can I do? And so the first thing I decided to do is to write a book about, essentially about water. And the purpose of the book was to make Gilbert Ling's ideas palatable. You know, he had a brilliant guy, but he didn't have a capacity to communicate his ideas as well as some others. And so I wrote this book with the idea of conveying his ideas to the general public. And, you know, among discerning readers, they will always pick this book out from among others. But you know, even for really bright kids, it takes a while to go through. Anyway, by way of introduction. So we started to do experiments because this seemed to be of huge importance to find out whether he's right. And I'll say at the outset that the general concept we were able to confirm, but there were some aspects that are wholly different from what Gilbert Ling was talking about, and these aspects I think are really important. So we started with the idea of Gilbert Ling's that inside the cell, that the water molecules shown here as dipoles, that is plus here, minus here, that dipoles could stack on one another. And in general, physical chemists would say that next to various surfaces, such as those inside the cell that are hydrophilic, that is, have charges on their surface, there may be one or two layers of water molecules that are ordered in the way. But Gilbert Ling was saying, oh no, they're wrong. It's actually many, many layers, hundreds if not thousands, and I'm later going to show you that it's actually up to hundreds of thousands, so it's a macroscopic effect. So we started with the idea of this kind of order, and when there's order, just as in a crystal, crystals tend to exclude contaminants or other molecules, otherwise they wouldn't be pure. And so we were looking for a region of water, a region that excluded molecules or particles. And we found it very, very quickly. We were looking for exclusion. And I'll give you a couple of examples of such. So here is a chamber. And in the chamber, we put water plus particles, and the particles were little spheres called microspheres, which are widely used in scientific studies. And we immersed a gel. It was similar to gelatin dessert, but it wasn't that. And immediately we noticed, or at a very short time, we noticed that there was a region that was actually devoid of these particles. And we thought, well, maybe, we had no evidence for such, but maybe this was a region that Gilbert Ling was talking about, where the molecules, water molecules were stacked in some way. But it was more dramatic than that, because watching it over time, this is one of the first videos, you could see that something was growing, something was pushing out these spheres. And as a result of that, someone, you know, we had many experiments on it, someone said, you should give it a name. And so we called this region of water the exclusion zone, or otherwise known as EZ water. And it works great in this country, but in Europe, EZ. This is easy to remember, but EZ doesn't quite work as well if you're in some other countries. But we began seeing this again and again. Later, we called this region because its physical chemical properties were completely different from ordinary water. We call this fourth phase water. So I use the term easy water, fourth phase water. They're equivalent to one another. Here's another example. This is a polymeric sheet of Naphion, which we use quite a lot, and water plus microspheres, again, and you can see the same sort of result. This region grows, and it can grow to, in this case, 0.5 millimeter, and there are cases where it easily grows to 1 millimeter. You can see it with your naked eye. So in terms of generality, what we found, to summarize, many hydrophilic, that is water-loving surfaces, generate these exclusion zones, and many solutes are excluded. I'd like to say all, but obviously we haven't measured all. So, next question. Is this water that we're talking about, this exclusion zone or fourth phase, is it really different from ordinary so-called bulk water? This is a summary constructed 10 years ago of observations made from our laboratory and other laboratories that easy water is physically different from ordinary bulk water. And I'm not going to go through these in any detail because otherwise it would take all my time. I've got other interesting stuff to present, but for example, this is the observation. Easy is more viscous. It's kind of gel-like. And the technique we use is in red, falling ball viscometry, et cetera, et cetera. Essentially, all of this is published. And if I were to include up to date, this is 10 years old, up to date, the list would be substantially longer. But I do want to talk about one of these observations, which I think is particularly important, that easy typically has negative charge. And we were able to observe this measuring the electrical potential using small electrodes that biologists have been using forever, at least 60 years or so, to stick into cells to measure the electrical potential of the cell. And what we found is schematically shown here. So here's the material with hydrophilic surface, and we found by sticking an electrode in here that typically it has negative charge. And of course, there had to be a region of positive charge elsewhere because we started with neutral water. And so it wound up that this was negative and the positive charges were here. And this is sustained over time, despite the proclivity of the positive charges to merge with the negative. And if I get to it, I'll explain the reason why these are maintained separated. But essentially, you have a battery because you have separated charge. And that's essentially what a battery is. And the way we found that it works is you start with water molecules that look something like this. And what happens is that energy, and I'll talk about the energy in a moment, splits the water molecule, as in the first step in photosynthesis, splits the water molecule into this negative moiety and positive moiety. And the negatively charged components gather together in this region to find this negatively charged region, and the positive charges are cast off into the periphery here. And thereby you obtain this charge separation. And we were able to deduce the structure of actually what happens in the EZ. And so it starts with material here and the hydrophilic material, and it starts with ordinary liquid water here. And what happens is that layers form one by one, and we found that these layers are actually hexagonal or honeycomb kind of structure, and they build one by one, as you can see here schematically. And if you look at one of these layers, you can see the hexagons here, the oxygens at the vertex and the hydrogens in between. And if you actually add in one unit cell, if you add the negatives and positive, the hydrogens and the oxygens, it turns out it's not H2O. And of course, it couldn't be H2O because H2O is neutral. And this was measured to be negative. And so if you do the count in a unit cell, it turns out to be H3O2. If you had H4O2, it would be double this, which is neutral. So you remove one of the positive charges, and thereby you get the negative charge in the exclusion zone. We were pleased to see a recent paper that shows this 3-to-2 ratio of oxygen to hydrogen. And if you just read down to the bottom, the ratio of hydrogen to oxygen in this paper, roughly 1.5 to 1, instead of the usual 2-to-1 ratio corresponding to a chemical formula of H2O. So I believe that what I've presented to you is true, and that there is a kind of easy water or fourth phase water that differs essentially completely from ordinary H2O that fills the cell. So when you think about the cell, the inside of the cell is really crowded. This diagram grossly underestimates the crowdedness of the cell. And the actual distance from surface to surface is equivalent to the diameter of about 6 or 7 water molecules stacked upon one another. The surfaces are really close, which means all the water is interfacial water, which means, since these surfaces are mostly hydrophilic, that it's all easy water that's inside the cell, as Gilbert Ling was suggesting, but the structure now is quite different from what Gilbert Ling had been espousing. And so the conclusion that we reach, or one conclusion that we reach, is that the negative electrical potential of the cell is due to easy water's negative charge. Now, I know many of you were educated in medical school, and you learned, just as I learned in the physiology course, that the negative charge of the cell had something to do with pumps and channels in the cell membrane. I've argued in various papers and in my last book somewhat that that's erroneous. And I think a simpler explanation is that the reason the cell is negatively charged has to do with the presence of easy water, which is negatively charged. So it's a very simple kind of observation that has really important implications that I'll get to. Now, what I would like to talk about next is information. When you think about information, just like the previous talk somewhat, about information storage and Moore's law and such, you think of a thumb drive and think about how information is stored. And I think many or most of you know that it's stored from transistors that are regularly arrayed in a two-dimensional format. And each of these has 2 different states, 0, 1, on, off, whatever. And it's this collection of zeros and ones, a distribution that stores the information. And I'd like to suggest to you that the structure of the EZ is pretty much the same, it has the same principles. It's not only regularly stored oxygens and hydrogens in a plane, but also in the 3rd dimension as well. And the issue is whether this has the capacity to store information. And I think there's good reason to think that that may be the case because of its similarity to ordinary memories. And the idea of water memory, which maybe is unfamiliar to many of you, but there are many scientists who have been studying this, and many of them have been discredited for their radical reviews. And the most prominent one was Jacques Benveniste, who had direct evidence for storage of information in water. And few people could believe his result, but his work has now been amply confirmed by multiple groups. He basically lost his career. He was a very prominent French scientist who was criticized, especially by the editor of Nature, and his career went downhill, and he passed prematurely, despondent. Another is someone who some of you may know. This guy was a spiritualist, not a scientist. And his work was doubted and still doubted by many scientists. But he would take some water and he would put his intention to the water. And he claimed that if his intention was positive, if he thought about peace, for example, or love, he'd look at the crystals that formed, and they were beautiful. And on the other hand, if he thought negative ideas, the crystals were ugly. And this is a kind of sample. So Mozart's symphony, John Lennon, beautiful crystals. If he was thinking about peace, thank you, he'd get crystals that look like this. On the other hand, negative thoughts, negative comments yield ugly crystals. And so few scientists are willing to believe these results, and maybe for good reason, because he would freeze 50 petri dishes and pick out the one that showed what he wanted to show. And obviously, no scientist would do that. On the other hand, we go to the opposite extreme, and here's Luc Montagnier, who won the Nobel Prize for discovering or identifying HIV and AIDS. And he was friends with this first guy I showed, with Jacques Benveniste, and he was so intrigued by those kinds of results that he essentially changed his career. And he would come to our conference, which I organize each year, the annual conference on the physics, chemistry, and biology of water. And I just want to show you one of his interesting findings. And we tend to think that if he was a good enough scientist to have won a Nobel Prize, that you kind of think that maybe his results are believable. And here's one of his main results. And if you just bear with me, so it has to do with water memory. So you put two flasks near one another. The flasks are sealed by methods better than illustrated here. And he put them near one another. And this one contained only water, pure water. And this one contained a short strand of DNA in an aqueous buffer. And his hypothesis was that information is going from the DNA or from the water that surrounds the DNA, and it's being passed to the water, not chemically, because there is no way for molecules to move from one to the other, because these were factory sealed. So they were next to each other for one day, and he would supply ambient energy in the form of 50 hertz or 60 hertz. And he was suggesting that some kind of information, be it electromagnetic or whatever, was being exchanged. And to determine whether that was true, he took container B, the supposedly pure water, and he used the PCR technique, which I think you all know about because it's been used so widely with respect to COVID. And he got an amplification of the DNA, and it turned out that the sequence of this DNA was the same as the sequence of this DNA, supporting his hypothesis. Now, of course, a lot of people don't believe this. It's now been confirmed and published by 3 different groups, 2 Italian and 1 Chinese. So it may indeed be true. So I've given you sort of the scope, one small segment of the scope of experiments that have been done to show water memory, which we don't think is possible, but apparently it's possible. So you can imagine now, reverting back to Moore's law, which I hadn't thought of until a few minutes ago. So imagine, instead of being filled with transistors, it's filled with easy water, which seems to be a substrate for water memory. We calculated that because we're speaking at the we're considering the atomic level, not the transistor level, that we can achieve an information density of something like 10 to the 9th over and above what we can do today. So we don't know if that's the future. But anyway, this is a you might say, a distraction from what I really want to talk about, which is health. If your cells are filled with easy water, that is the default state, which means we're healthy. But I'll get to that in a moment. So I just want to summarize to answering the original question: is easy water physically distinct from bulk water? And what I've shown you, I think the answer is yes, it is. It's as best we understand, a layered honeycomb structure that has information storage capability. And it fills our cells. So now I mentioned to you earlier that this is because of the charge separation. This is like a battery. And the question is, what charges the water battery? And the answer turns out to be very simple. It's light. Light. And, this is not a conception that arose from my brain. I had been scratching my head for many years. That's why it's a little thin here. And we couldn't figure it out. You can't take the chamber and plug it into the receptacle to get energy, but you need energy of some sort to create a battery, to charge a battery. And it was a student, a young undergraduate student, who found the answer. He was sitting or standing at a bench doing one of these experiments similar to what I showed you early on. I think he was bored. And so he noticed sitting right next to him was a gooseneck lamp. He took the gooseneck lamp and he shined it on the chamber, and he called me in. And this was actually his original result. This is obviously an artist's conception, but so he had a hydrophilic material, Naphion in his case, and generally, you can see the exclusion zone here and the microspheres here, and generally the boundary between these is parallel to the boundary of the nucleating material, but where he was shining the light, it was growing. And so he called me in, and I was amazed to see. And of course, the first thing we did was to repeat the experiments, and we wanted to find out which wavelength of light was most efficacious for all this, because the lamp contained a series of wavelengths. And what we found is that the most effective was infrared radiation. The UV light had very little effect, nor did visible light, except when you get toward the reds. And when you go beyond the reds to the infrared, particularly at 3 micrometers, it was a huge effect. And we found we could build it up easily using a weak LED up to 10 times the size. So it didn't take a rocket scientist to figure out that the energy was coming from light, and particularly infrared light. And many of us are not so familiar with infrared light. We know that the sun contains a lot of infrared. Roughly half the radiant energy from the sun is in the infrared region, but infrared is all over. If you turn off all the lights, take out your infrared camera, which is the same as the ordinary camera, but the sensor is sensitive to infrared wavelengths instead of visible wavelengths. You get a beautiful image, and that's why military uses it in darkness to spot the enemy's tanks and such. So the energy is all over the place, and therefore, if you have a hydrophilic material sitting next to water, since the energy is available to propel the conversion, you have a fair amount of easy water. And if you were to add more infrared energy, then it grows. And if you take it away, it returns to the default. So energy for buildup, we found, is photonic energy. It's light that does it. It charges the water battery. To get the energy, you can't invent energy. You can only get it from another kind of energy. And it orders the water. So effectively, it's very simple. Just lie in the sun. It contains plenty of infrared energy and basically recharges the battery. Now, if the battery gets recharged so simply, just by being in the light, you'd think, or you'd raise the question, is this energy, can it be harvested? Can you actually use this energy for some useful purpose? And the answer is yes, and the answer, in fact, came once again from a young undergraduate student. He was actually a freshman. We discussed a project, and the project was, we found that Naphion exists not only in sheets, but also in tubes. And I asked the student, please, can you check to see, because of the curvature, does it really generate exclusion zones the same way as flat surfaces do? And he went to work, and quickly, while I was out of town, he found, yes, it does. And, you know, he had nothing to do. And so he kind of thought to do something more than just confirm that exclusion zones did exist both outside and inside. And so I came back from my trip, and I'm sitting in my office having a discussion with some important guy who turned out to be not so interesting. And this student, you know, usually the students are polite. I leave my door ajar so they can feel free to come in. But usually, you know, are you busy? And I was sitting with this guy, and the student walks in, he barges in, which was uncharacteristic, and he says, I found something that I thought might interest you. Okay, so, you know, I wasn't expecting a whole lot, but what he found was really astonishing. So he came in and he told me, he says, you know, I'm looking at the tube underwater, and I noticed that flow is going through the tube, and it just keeps going and doesn't stop. And I'm thinking, something's wrong here, or something very interesting, because usually you need a pressure gradient to drive flow through a tube, because water has viscosity. And there's no pressure gradient because there's no difference in height between here and here, but yet it was flowing. So we quickly checked to see if it was correct, and it was correct. And the way it works is this: you fill the inside of the tube with water, make sure there are no air bubbles, and you immerse it into water plus microspheres, like that. You take it to the microscope, and look inside, and here's what you find. So here's Naphion, exclusion zone next to it, and you could see the flow is going through. We had it going up to 1.5 days without stopping, and we know how to make it longer. Well, someone came in and said, oh, you know, I'm not sure that I believe this, because there may be some special characteristic of Naphion that's unusual. And so he prompted us to look elsewhere. We couldn't find small tubes that were hydrophilic. Maybe it's not our field. So we created one. It was actually another student who created one. So he took a gel, but before it gels, it's a liquid. And as it was gelling, just like Jell-O, he took a wire and he stuck the wire in. And just as it was gelling, he pulled the wire out, so it leaves a chunk of gel with a tunnel. So we took this small chunk of gel with tunnel, we immersed it in water plus microspheres, so here's it was polyacrylic acid gel here, and here's the tunnel. And as I was showing you earlier, EZ builds next to the surface of the gel, here too. And here are the microspheres confined to this region, and we found the same result. It flows. We tried this with 6 different gels, and the result was the same. So this is general. Of course, the velocity was different, other characteristics were different, but basically that's what we found. Another person said, well, you know, you think this is powered by light? Why don't you just add light? So we turned up the microscope light to get more, especially in the infrared, and we found that we could actually get a speed increase up to, I believe it was 5 times higher. We published this result. So the bottom line of this is that work is done, and therefore energy is required. And the only energy that we could conceive of is the energy absorption that comes from the infrared that's around all the time and gets absorbed by the water. And somehow this is getting transduced into mechanical energy, light energy getting transduced into mechanical energy. So the question that arose to us now is, does human biology use radiant energy? Do we use light? We don't think that we use light, but I want to show you that I believe we do use light. So there's Mother Nature, and Mother Nature was quite successful in using light and green plants and some bacteria. And if you were Mother Nature, would you continue to exploit this kind of energy after you invent animal life? You could say, well, animal life can forage. But on the other hand, a more conservative view would be, yeah, well, maybe use it as a backup or in conjunction with other kinds of energy. So the first issue that we thought of was that we're receiving energy all the time, and especially the cardiovascular system, the capillaries, are pretty near the surface. And so one idea is maybe this energy that's coming in all the time is used in the cardiovascular system. Of course, we would never think of such a thing, and I would never think of such a thing if not after a visit to Moscow University to my friend Vladimir Bayakov from the biochemistry department. And one visit there, after we sipped some vodka and such, he took me to his lab and he introduced me to his colleague from down the hall. He has something interesting to tell you. So we go to the colleague, and the colleague tells me, he said, there's a problem in the cardiovascular system. And I'm thinking, I did my PhD simulating pressures and flows in the cardiovascular system, and I thought we had all the answers. So I must admit, my nose was cutting the clouds. I felt pretty arrogant, but he had me convinced that he was right within 5 minutes. And here's the issue. The issue is that the red blood cells are 6 or 7 micrometers in diameter. The capillaries, the small ones, are half that diameter. They're 3 or 4 micrometers in diameter, which means that for a red blood cell to pass through many or most of the capillaries, they have to get squeezed. And you can see this is a muscle. You can see the striations, and here you can see the capillaries. And I'll play the video for you. You can see they have to get squeezed. They get bent, and this guy gets awfully bent and having a hard time getting through. He said the energy that's needed to bend, he did the calculation. He said that if the left ventricle were responsible for creating enough pressure to do this, the ventricle would need to generate a pressure of something like 1 million times the pressure it actually generates. That's high blood pressure. You can see the problem here. So the red blood cells are supposed to look like here, but you can see that they get definitely squeezed. So he said there must be some other source of energy that's driving these red blood cells. And I don't have a slide to show you, but my student checked to see something really interesting, that scientists, a half dozen different scientists over the past 100 years found something really critically interesting, that if the heart stopped, the flow continued at low velocity, but it continued, supporting the point of view that this Russian guy was talking about, that there must be some other source of driving energy. So I was thinking, since we had just found the flow through the hollow tube, I was thinking, is it possible that the same phenomenon existed in the cardiovascular system, and that the blood vessels themselves were subject to this phenomenon of flow based on radiant energy coming through, the same flow mechanism that we're talking about. Might radiant energy help drive blood flow? And my student did the experiment, and he told me after he had a successful result that when I suggested this project to him, he thought I was on some kind of drug. changed his mind afterward. Okay, so he did the experiment, and he used a chick embryo, 3 days old. And at 3 days, the cardiovascular system, which you can see here, is pretty well developed, but the regulatory systems, the hormonal and neural system, were not well developed. So you can see the egg here, the top is lopped off, and you can see the cardiovascular system. And he developed an imaging system to look at a couple of different vessels. And the first thing he did was to stop the heart, which you can do by injecting potassium chloride. And after he stopped the heart, he confirmed that there was flow to start with. So here's his main result, which was just published a few months back after multiple rejections. Here's blood flow versus time. So the first thing is you stop the heart. And the flow starts at some high value and goes way down, but it doesn't go to zero. It goes to some value. And then he tested the signature feature of our phenomenon. We saw in the laboratory that it's run by infrared energy. So we applied infrared energy here, and you can see that the flow increased by about 3 times, and then stopped the energy, and it decreased down to some baseline level. So we were able to confirm that radiant energy does help drive blood vessel. And what this means, if you think about it, it means that in your cardiovascular system, and maybe mine, it's not just the heart that pumps the blood, it's the vessels that pump the blood as well. We, because of a technical issue with this experiment, we can't tell or couldn't tell what percentage is driven by the heart and what percentage is driven by the vessels. But we confirmed that it appears that in your cardiovascular system and mine, it's not just the heart that does the pumping, it's the vessels that do the pumping as well. So we're using energy, light energy, infrared energy, that's available in the environment, and also getting generated by the core of our body, which generates heat. That we actually use this kind of energy just the way plants do, maybe not exactly the same way, but pretty much similar, so that nature does use infrared energy is demonstrably in our body. How exactly does it work in a more generally? Well, remember I said the cell is crowded, that negative EZ practically fills the cell. And the way potential energy is actually used by the cell and is that the cell is filled with negative charges. The negative charges repel one another. They want to get away from each other as much as possible, and that constitutes potential energy. And that potential energy is used to drive protein folding, which is essentially what drives pretty much all the processes that are undertaken by the cell. And I discuss evidence for this in my Cells, Gels, and the Engines of Life book. So it goes something like this, that you've got a protein, and the protein is going to fold, and the protein is surrounded by EZ water to start with. And so what happens is, step one is that the EZ water melts into ordinary water, and the protein undergoes folding. Then the last part of the cycle is it returns to where it was, and EZ water builds up. And actually, that's the energy requiring step, the build up, the return of EZ water to this initial state. On the other hand, if you don't have EZ water, and what I really mean is there's a deficiency of EZ water, then the cell is basically impotent. It can't do what it's doing, and the cell becomes dysfunctional or even pathological. So essentially, if you connect the dots, basically the potential energy from EZ drives work. So the buildup of EZ is an energy source that we use necessarily beyond the cardiovascular system. If you trace the steps, remember, light builds EZ, which has negative charge, which has potential energy, and that potential energy is used to propel protein folding. And if you connect light, yields work. So what that means is essentially that we use light energy to do work. It's not just the chemical energy that is discussed in so many textbooks, but light energy in pretty much the same way as plants do. We use light energy as well. So if you ask the question, well, where do we get our energy? Of course, we get some energy from food, but we also get energy from light. The light is absorbed by the water, and I can't read my writing, and that is used to supply energy. And so if you think about people like Prahlad Jani, which I'm not sure if many of you know about, this guy claimed for 65 years to have had no food and no water. He lives in a humid place, or lived in a humid place. And the question is, and he was examined by a group of 15 physicians who certified that at least for the 2 weeks where he was in the hospital room, he had no food at all. And afterward, he was able to run up the steps to the office of the guy who was in charge of examining him faster than the doctor could run up the stairs. So this guy was getting energy from somewhere, and if you think about it, if it's not from food, it's got to be from somewhere else. And the suggestion is that it may be from the infrared energy that he was absorbing. And so so-called breatharians, there are said to be something like 200,000 of them around the world. It's not rare. Where do they get their energy? It's possible that it's light that's absorbed by water and turned into potential energy. So for building energy and creating function, should this all matter to you? Well, light matters and water matters. And the question is, well, what can build easy water in your cells if easy water is necessary? And I'm going to list a half dozen expedients that could be important to you when you think about health, if all of what I said, or some of what I said, is true. So the first is to drink a lot of water to remain hydrated. And I don't think too many people would disagree with that because water is the raw material for building easy water. You take in water, you have a drink, and the infrared energy then converts the sum of the water, at least, into easy water, which you need, absolutely. Green juicing. You go to your backyard, and you take some of the freshly grown green leaves, squeeze out the water. So what is the water that you're squeezing out? It's the water from inside the cells of freshly grown plants, full of easy water. So you're actually drinking easy water. And many health practitioners tell me that this is the simplest expedient for improving health. That's been their experience. It's full of easy. Another is that there are various substances that have been known since Ayurvedic times to be good for health. And we wondered whether the reasons these substances are so good for health, a wide range of health applications, is maybe that they build easy water. We tested half dozen of them, like turmeric, coconut water, basil, holy basil, and various others, and we published it. It turns out it's true that in doses that are similar to what we might be taking, they build easy water, all of them, and we published that. Sunshine. In Seattle, where I live, in the wintertime, we don't see much sunshine. When the sun comes through, people are smiling. And the effect is assumed to be a kind of psychological effect, and maybe that's true. On the other hand, sunshine is full of infrared energy. The infrared energy penetrates through our skull, it demonstrably does, reaches our brain, and in the brain cells builds easy water. It might be that we feel better when the sun comes out because we revert to the default state, which is feeling happy, not feeling depressed. An extreme example of that is the sauna, or as the Finns say, sauna, and it's heat. And heat is essentially the same, not exactly, as infrared energy. So you go into a sauna, you feel depressed, full of pains, and you come out 30 minutes later and you're feeling a whole lot better. I had the same experience myself. And basically the sauna is just filled with infrared energy. And I think that could be the reason. And finally, grounding, or as the Brits say, earthing, connecting yourself to the earth. I originally studied electrical engineering, and no professor ever even hinted that the earth was anything but neutral. And one day, a Russian colleague who was in my laboratory working, he was about to take off for Russia again, and he starts talking about the Earth's electric field. And I said, Andre, you must be talking about the magnetic field. I never heard of an electric field. He says, you never heard of it? I can't believe it. He said, every Russian middle school student, and I confirmed this was true, every Russian middle school student, at least 30 or 40 years ago, knows that the Earth is negatively charged. And I went home, and my head was spinning. I couldn't believe that this could be true. And next morning, one of my students brings Feynman's lectures, volume 2, chapter 9, filled with evidence that the Earth is negatively charged. So there you go. Okay, so I know we're running short of time, but I have to address one more issue briefly, and that is an issue that that interests all of us, and that is the issue of cancer. And I think you all realize that during the past 50 years, starting with President Nixon's war on cancer, which obviously has not been won, it's mostly been the geneticists who have dominated the field. But I'm thinking in a somewhat different way, and I asked the question about what prompts cells to divide, because that's characteristic of cancer cells. And I went back and I found studies by a guy named Clarence Cohn 50 years ago. And what he found is that if you take a cell and you reduce the electrical potential to something close to zero and hold it there for a while, the cell will divide, any cell. And if you did the opposite and you raised the electrical potential, raised what I mean is the magnitude, to instead of minus 10 or minus 20 to minus 80 or minus 90 or 100, the cell would never divide. And I thought, well, this is pretty interesting, because we know something about the electrical potential now as being the result of negative charges from easy water. So recently I wanted to find out how general this was. I looked at a review article, and this is from 2013. Maybe there are more recent ones. And so what this shows is the so-called membrane potential, low potentials here, low magnitude potentials, and high magnitude potentials here. Tumor cells in this category, non-tumor cells in this category. And if you look at the proliferating, that is dividing cells, some dividing naturally and some dividing because of pathologies, they all have small electrical potential. And if you look at the non-proliferating cells, like glia cells and neurons, skeletal muscle, they have high electrical potential. So this seems to be general, that cells will divide if they have low electrical potential. And tumor cells are exactly in that category, very low electrical potential. So the low electrical potential means division. So in cancer cells, they all have low electrical potential and they all divide. And therefore, what you need to do, the logic follows from that. The reason they have low electrical potential is the amount of easy water in the cells is minimal. you need to build up the electrical potential. And the buildup of electrical potential should reverse the division of cancer cells. So a therapeutic approach that follows directly from that is to build easy water, have more easy water in the cell, and that should build the electrical potential. And so an anti-cancer approach would be the same approach as that half dozen list, but there's actually more of them, same approaches as described. So in other words, if we're on the right track, it means that you can reverse cancer by building easy water, by using all those expedients that I listed. And that should return the cancer cells and other pathological cells to their natural working state. And so if we're filled with easy water instead of bulk water, we should be healthy. So I end with practical applications because I know a lot of people are interested in practical applications. And one of the things that follows directly from what I presented is getting energy from water and sunlight. You've seen this before. So the idea is you put one electrode here in the negative, one electrode in the positive. As I suggested, this is a battery, and you ought to be able to get electrical energy from that. We've demonstrated proof of principle. And it's demonstrated here in the slides. So here you see a series of about a dozen chambers, and each chamber has some naphion in it and two electrodes, one in the negative zone, one in the positive zone. And the chambers are connected either in series or in parallel. Here's a switch. Here's an LED lamp sitting here and a magnifier. And so you flip the switch, and you can see that the water is producing electrical energy. So we don't know in the future whether this has practical value, but it may well. It's essentially water and sunlight giving you electrical energy. And whether it can solve the energy problems of our world in the future remains to be explored. We haven't done that yet. Another is getting drinking water from contaminated water. And we have shown here a kind of filter. We call it filterless filter because it has no physical filter. So from what I've shown you, here's a tube. It could be a naphion tube or any hydrophilic material. And the inflow consists of water plus contaminants. It could be pharmaceuticals, it could be microplastics or whatever. And as I've shown you, you get an exclusion zone here and here, and all the junk moves to the center. We built a device which has a fancy name, differential extractor. It's merely two concentric tubes, and all the junk is right in the center. We collect it, and it gets dumped here. And then the peripheral area is basically easy water, and you can see the result here. And we've been able to achieve in one single pass a 200 to 1 separation. So, and we have actually worked on the development of this somewhat, this filterless filter, and it looks promising. Even more promising is the possibility that we can actually separate salt this way. Some of us had a tour yesterday, and you can see the complexity of the apparatus that's used and the amount of electrical energy that's needed. This needs no electrical energy. It's built on the energy from the environment. And the idea is that salt is treated as a so-called contaminant. You can get rid of the salt and dump it here, and here have desalinated water. We don't know if this will work. We have some preliminary evidence. It remains to be seen in the future. So I conclude by saying, I guess the main central topic this is that we all have learned that water has three phases: ice, liquid water, and vapor. We found a fourth phase, and the fourth phase, we put it here between the ice and water because the fourth phase is hexagonal. Ice is also hexagonal, and it turns out, experiments which I have no time to present to you, that if you want to freeze water, you can't go directly from water to ice, you must go through the EZ phase, which then goes to ice. And if you want to melt the ice, you can't go directly from ice to water. You go from ice to EZ and then to water. So that's why I put the EZ here between ice and water. But essentially, there are 4 phases, not 3 phases. And there are broad implications. That the main implication is that water is absorbing radiant energy from the environment and is transducing that energy into other forms. And I've demonstrated to you that one of these applications is blood flow, that we presume that the flow of blood is driven only by the heart, and I've demonstrated to you that it may be that it's not just the heart, but the vessels themselves, which could be presenting an appreciable amount of energy driving the blood flow. In terms of chemistry, we've all learned from the chemistry books aqueous reactions, but these mechanisms for aqueous reactions are all built without the concept of charge separation, energy from the environment. And if what I presented to you is true, or is partly true, it means that in the chemistry textbooks that many of the interpretations will need to be changed fundamentally. Weather, we all want to know what the weather is going to be, and especially in Seattle, when we see gray skies in the winter, we don't know. We got 50% probability that it's going to rain. It doesn't help us. We don't know whether to bring an umbrella or to work or not. What's missing, I believe, in these forecasts is the fact that clouds are charged. We know that thunder clouds are charged, but it's not just thunder clouds. All clouds are charged. That's what, by the way, keeps the clouds afloat. And I believe that if we take into account the kind of water that's in the clouds, easy water, it should be easier to understand how to predict the weather. And my next book, which is coming out in a few months, contains 4 chapters on that, which start with the fundamentals of physics and chemistry and build up to an understanding of hurricanes and the like. So another is health, and I've touched on the issue that the easy water, feeling ourselves, is absolutely central to health. In terms of food, if you want to preserve food either by dehydration or by freezing, it's nice to know something about those processes. And from a so-called practical point of view, filtration is one issue that comes directly from that. Desalination is a possibility, needs to be explored, and getting electricity from water. So many of these are described in this book, The 4th Phase of Water, which turns out to be fairly popular. You can check on Amazon. So I think I'll end here. Thank you very much for your attention. Okay.